Hello, everyone. Welcome to Foresight Space Group. And today we're so lucky to have Chris McKay here with us. Chris, it's been maybe 2 years that we've been looking to have you on the group. So It's so wonderful to have you. And I will send it over to Creon to give a brief intro.
Hey, yeah, Chris and I both worked at NASA for many years. We never actually worked together, but we saw each other socially and we had a lot of friends in common. And I worked with some of them, like Larry Lemke, who sometimes comes to these meetings. And was always an admirer, of course, of Chris and tracked his work from afar. The work that spans a broad range of stuff and broad career: geology, geochemistry, planetary atmospheres, solar system science in general, and particularly with the focus at sometimes of life in the solar system, life on Mars, life on Enceladus, life on Titan, potential life on Venus even, and life on extreme environments on the Earth, as well as Mars missions, human missions, terraforming, all kinds of things. It's a very interesting guy with a very interesting set of accomplishments and positions. And I am now going to hand it over to Chris to take it away.
Great. Thanks, Creon. And so without further ado, I'll just get started. What I'm planning to do is talk about life on Mars. I'll try to explain why it's interesting and what's going on and what the questions are. And then I hope to have a lot of time for questions, because for me, that's always the interesting part. Since I already know what I'm going to say, it's rather dull. Okay, so let me see if I can figure out how to share. I think I've got it. So tell me if you're seeing my screen. You should be.
We're good. We got your whole slide presentation.
All right. I think when I go to presenter mode, I don't see you guys anymore. So if occasionally, Creon, you just grunt or say, yep, just so I know that there's still a connection and I haven't lost internet and I'm speaking to the vacuum. Okay, continue to share. Okay, so topic life on Mars, that's today. That date has changed a lot. Creon and I have been trying to organize this, and for one reason or another, the dates just didn't overlap, and they almost didn't overlap this time, but we got it. So it's a pleasure to talk to you. As Creon said, we've been trying to organize this for a while. What I want to do is talk about what I call the Mars questions, all of them related to life, in my view, and I group them into three: past, present, and future. And the first question, and I want to remind you, these are questions. In fact, I ought to put question marks after these statements. It's a question. Did Mars have life? And if it did, was it a second genesis? Question. And I'll explain what I mean by a second genesis. The second question is, can Mars support life? Is it a place where humans can live and work? I want to emphasize that this is a question because there's a lot of talk, loose talk, that assumes that the answer to this is yes, and that we don't need to even ask the question. But we'll come to that. And finally, the life future. Can Mars have a biological future? Again, a question mark. Some people talk like we know the answer already, but we don't. We have to do work to find the answer to all of these questions. So I'll start in the past, search for a second genesis of life. Notice I say a search for a second genesis of life, not a search for life, because I'm really not interested in finding life on Mars if it turns out that life is just part of terrestrial biology. It's like finding marsupials in Australia. That's interesting. It's a paper or two, but it's really not a fundamental breakthrough in our understanding of life in the universe because they're related to us. So what's a second genesis? The easiest way to understand it is to look at this diagram, the tree of life. It shows that all life on Earth shares a common phylogenetic and biological history. It's one life on Earth. There's one life as we know it. And it traces back to one universal common ancestor, last universal common ancestor, this little root on the tree here. These are the bacteria, the archaea, and the normal life, trees and plants, animals and things like that, the eukarya. If we found the second genesis, why is that important? Practically, we now have another type of biochemistry to compare to, Life 2.0. And if you think about it, many of the most important implications of science for human life root in biology: agriculture, medicine, pest control, whatever, reproduction, everything you want to think about that's really important to you. If you trace it back, it probably goes back to biology, or in one sense or another, biochemistry and biology. So having another example could give us profound insights. And when pressed, I use this example. Look at this tree of life. Here are the two microorganism branches, bacteria and archaea. They both live in our bodies, in your bodies. more bacteria than archaea, but there's untold billions of them in your body, maybe millions, but billions of one sort or another. And yet only the bacteria, cause disease. There's no pathology associated with any archaea, which isn't that curious. There are pathologies associated with eukaryotes in your body called cancer. The eukaryotes go crazy and they grow, and the bacteria go crazy and they grow. But the archaea in your body are always well-mannered and they never get out of control. And why is that? We have no idea. And many years ago, I emailed my smart biological friends and I said, smart microbiological friends, why are there no pathogenic archaea? And they wrote back, dumb planetary scientists. The reason, obviously, is that archaea never developed pathogenicity. Oh, that explains a lot. The more reflective microbiologist I asked, he said, there are pathogenic archaea. We just haven't discovered them yet. Very interesting. Maybe it's got something to do with the nature of the tree of life. And maybe only by finding other trees can we understand this tree.
Did the archaea just come about so early that there was nothing to attack and pathogenize?
No, archaea are just as old as bacteria. In fact, the tree of life is a little bit over here, and archaea and eukarya share more in common than eukarya and bacteria. So your question is complicated. I gave you a very simple-minded answer, and people are working on this deeply ever since Lynn Margulis and pushed the endosymbiotic theory for the origin of the eukaryotic cell. It's a whole field all of its own. So what we're looking for in a second genesis is not on our tree of life, aliens. And an important point here is we define aliens. I define aliens not geographically. You're not an alien depending on where you come from. I don't care what the politics says. You're an alien if your biochemistry is different from our biochemistry. So if we go to Mars and find life and it maps on our tree of life, it's not alien. If we find something in our backyard that's completely different than anything we've ever found before, and it does not map on our tree of life, it is an alien. We might have aliens in our backyard, never found them. So it's, in a sense, my job description is searching for aliens on other worlds. I sit down on a plane and the guy next to me wants to chat. He says, what do you do for a living? I said, I work for the government and I search for aliens on other worlds. And boy, that ends the conversation right there. And I get a nice quiet flight. And I sometimes get a whole row to myself. Anyway, more seriously, this is important. It would tell us not only comparative biochemistry, but that life is common in the universe by the 0-1 infinity rule. Right? There may be only one type of life in the universe. We know it's not zero. But if we get to 2, that immediately tells us that it's up there in the billions. Okay, so why Mars is the target? Obvious answers: evidence for past liquid water, presence of an atmosphere, showing that it's got the elements needed for life. And importantly, it's been cold and dry for a long time. So there's a good chance that if there was evidence of life, it's still preserved in the freezer. Okay, so that's why Mars. Why isn't Mars habitable? To simplify, it's because it's too small. If Mars started out as we think, with a warm Earth-like period, the lack of plate tectonics, less gravity, and no magnetic field caused it to lose its atmosphere and become the cold, dry world. And there's debate on the relative importance and timing of these effects, but there's a general consensus that, yeah, Mars is too small. It doesn't have the properties that could maintain its habitable state the way the Earth does. Okay, the one problem I always have with these artist conceptions is they make Mars look like it's Miami Beach, even when it's 3.5 billion years ago. And there should be more ice in these pictures, as I'll come back to later. We know Mars had water. We see water frost on the surface, evidence from past water in terms of fluvial features carved in the surface. There's geological evidence for lots of water in the past, and there's evidence for water on Mars today, but not very much, not as much. In 1976, we're coming on to the 50th anniversary of the Viking mission, which was the first and to date the only mission to search for life directly on another planet, and that was on Mars. The Viking had 3 biology experiments. The details here are not that important, but basically what it did was take soil and add water with soup, add soup to soil. You add soup to soil and see if there's anything in the soil that would eat that soup. Remember, these experiments would have been designed in the late 1960s. Okay, and 3 biology experiments. It also had an instrument that sought to detect organic material directly. The key results, I'm not going to go into this detail. This is more for the science background. For those of you interested, these charts will be posted, I think. There was reaction. Stuff happened that surprised everybody. Oxygen was released from the soil when it was wetted. Something ate the soup. There's something in the soil that consumed the soup. And there was no organics reported. Puzzling. And there was lots of arguments over what this means. The folks that measured the reactivity said, this is life. The folks that measured oxygen said, this is bleach. And the folks that measured the lack of organics said, this is nothing, right? And they just were at each other's throats for many years. And I'll come back to now what I think is the resolution of this paradox. Looking back at the Viking results, an important point is we would not do it that way now. Viking tried to detect life by growing organisms, more, in other words, by feeding food to organisms and seeing if they would eat it. It's not a good sign of life. If any of you have kids, you can feed things to people and they don't eat it. It's uptake of food is not a very good indication of life. And in fact, we now know that 90% of soil bacteria won't grow in culture. The Viking biology experiments would not have been able to detect 90% of the soil bacteria right here in your own backyard.
And by the way, Chris, isn't your claim or claims from your school that you could have landed Viking in the Atacama Desert and gotten the same results of no life?
Right.
On Earth?
You would have, if you'd taken that, and that's because of this final point, that there in the Atacama Desert, there's locations which have organisms in them, but that none of them are culturable. You can take that soil and put it in a variety of culture media, and nothing will grow. And yet we know by direct DNA extraction that there's life there. So on Earth, we don't do growth experiments to search for life. We do DNA, it's direct. So we're going down to the Antarctic in a few months. Team is going to be collecting samples, talking to the microbiologists. They say, we need a lot of samples because the DNA is so sparse in those soils. We got to concentrate it. They wouldn't have a chance of detecting life the way Viking did. But with DNA analysis, we can. But if you're looking for an organism that doesn't have DNA, you're out of luck. Okay, but we'll get to that later. The whole picture changed radically in 2008 when Phoenix landed on Mars. This was probably, or certainly in my opinion, the most important mission after Viking, Phoenix. It went to the polar regions, landed on ice-cemented ground, and found ice-cemented ground as expected, but it found something that was completely unexpected, that completely changed our view of what Viking did. So what Phoenix found was ice. Yeah, okay, it was targeted to land on ice. The soil was slightly basic. There was carbonates, but there was a unexpected presence of perchlorate. I didn't even know I was part of the team, the science team. I didn't even know what perchlorate was. Now it's my favorite molecule. Perchlorate is a stable, highly oxidized form of chlorine. It's a chlorine atom with 4 oxygen atoms, like a methane equivalent with chlorine and 4 oxygen atoms. And it's the most oxidized form of chlorine. You don't find it very often on Earth. You find it in places like the Atacama Desert at very low levels, but you also find it in solid rocket boosters. People who are familiar with model rockets, they know all about perchlorate. Perchlorate is used as the oxidant in model rockets because when it's heated up to high temperature, it releases very reactive states of oxygen, which burn the fuel and make the rocket go up. The space shuttle, the solid rocket boosters on the space shuttle were packed with perchlorate. It's also, interestingly, can be consumed by microorganisms. They can use perchlorate the way we humans use oxygen, right? Humans eat organic material, preferably chocolate, and they react it with oxygen, and that gives them energy. Microorganisms can take that same chocolate and react it with perchlorate to get energy. For humans, though, perchlorate is toxic. It causes a thyroid condition that the levels it's seen on Mars are thousands of times higher than the levels regulated by the EPA, although that might change under the current administration. Okay, organics on Mars, post-Viking, now everything we thought we knew from Viking is wrong. The upper limit, the immediate prediction of perchlorate in the soils, what Viking saw, what caused the reactivity, what did not allow them to see organics was perchlorate. The prediction was that there would be perchlorates widespread in the soil on Mars, and there would be organics. Viking didn't see them because the perchlorates destroyed the organics in the chamber as they were trying to detect them. And in fact, Curiosity 2012 landed, and that's exactly what it found. Lots of perchlorate and organics that could be detected if you found a way to see past the perchlorate, which it did. Okay, now I want to talk about what we're doing next. And part of it is based on searching for life in places on Earth where there's not much life at all. This is where we get lessons on how to search for life. And I want to talk about two places just to give you a sense of it, both in Antarctica. Not surprising, although we work in other deserts too. This is the highest, driest valley in Antarctica, University Valley, one of the highest, driest, if not the highest, driest valley. It's 1.5 kilometers high. The mean annual temperature is -20 centigrade, -22. The air never gets above freezing. There's ice, is never bulk melting, dry, frozen ground with ice, cemented ground below, just like we see on Mars, but there's life there. That's a place where we try to test our instruments that we want to send to search for life on Mars. Perseverance is going to a different type of site, landed in 2021, started as going to the same type of site that Curiosity went to in 2012, which is a big lake bed site. Here is the lake that Perseverance went to. It landed at the Red X. This is an artist's conception of what that lake looked like 3.5 billion years ago. If we had gone there 3.5 billion years ago, we would have splashed down in this lake. But actually, that's not correct. We would have hit the ice cover on the lake. As I said, artists keep forgetting the fact that even back when Mars was wet, it was likely very cold. And so this lake would have probably been an ice-covered lake. But that is not a fundamental problem for life, as I'm going to show you now by taking you to places. This is another picture of the Jezero crater. Looking out, you're seeing the sediments and rocks in the hillside. You're standing on a dry lake bed. Billions of years ago, this would have been deep underwater. Now it's dried out. So these are lake sediments mixed in with other stuff. We find environments like this in the Atacama Desert, in the Antarctic Dry Valleys. But the most relevant analogs, I think, are the lakes that are found in the coldest, driest places on Earth. And this is, of all the lakes, my favorite one. So the one I'm going to show you. This is a lake in East Antarctica called Lake Untersee. And this is our camp there a few years ago. Just to engage in the political discussion today, this is our facilities, and they are not gender specific. You're allowed to use them regardless of your gender. You're required to use them regardless of your gender, self-declared or biological. This is number one. Number two is around the bend in a little box over here. And the view is great, the conditions not, but the point here is an important one, which is every drop of water that we touch, every drop that we use in cooking, every drop we use in brushing our teeth, every drop of waste, urine and solid waste is carried out with us. Our motto is leave no organics behind. And we don't. And the reason we don't is because the organic load in our camp is equivalent to the organic load in this ultra oligotrophic lake, probably in thousands of years production. So we can't contaminate this lake biologically because nothing that lives on us would grow in this water, but we could contaminate it organically, which is why we carry out everything we bring in with us. Okay, so we're testing out the hypothesis that in an otherwise lifeless environment, cold, dry, lifeless, underneath a cover of ice, there can be a thriving ecosystem. And the ice here is thick. It's 3 meters thick. So we take standard Jiffy drills that are designed to go through 1 meter of ice, and we just string them together. And you see me here reaching up. And my main qualification for being on this team is I'm taller than anybody else on the team. So that's my reason for being there. We drill holes, take samples, and do the standard limnological analysis. But we also did something that had never been done before, which is dive into this lake. We cut a hole, an access hole through the ice, and then we send non-essential federal employees down into the ice to look and see what's there and take samples. And it's amazing to think that we Our team was the first humans to get in this lake, but not just the first humans. We were the first vertebrates. We were the first vertebrates to go into this lake. This lake has only microscopic things like diatoms and algae and cyanobacteria. We were the first thing with the first invertebrates to enter this lake. It's been isolated for that long. And what we find in this lake is a purely microbial ecosystem, purely microbial for reasons of isolation, not for reasons of chemical extremes, which is where we often find microbial dominated ecosystem. Isolation and living alone, these microorganisms do things they never do when they're sharing their ecosystem with larger organisms like fish and frogs and amphibians, which graze on them. They develop these huge mounds, huge by microbial standards. These are the microbial equivalent of pyramids. They are thousands and thousands of times bigger than the individual organisms that make them. These mounds are about 30 centimeters high. And they are, we think, living examples of the kind of mounds that provide the earliest evidence for life on Earth. So our question is, could this lake on Mars have looked like this lake in Antarctica 3.5 billion years ago? Why not? It's very cold. The mean temperatures are probably not too different. And could there have been life on it? If so, it would have been the sort of microscopic life we see here. And so the sediments we see at Jezero Crater and these other equatorial lake beds on Mars, these are lake deposits. And if there was life, they're going to be evidence of that's going to be trapped in these lake deposits. So very exciting to get those sediments and study them. And that's what the Perseverance sample returns are supposed to do. Okay, I now want to address the question, what if we succeed? What if we bring back organic material on Mars, back to Earth? How can we tell if it was ever alive? As I said earlier, the first thing you'd probably want to do is look for DNA. That's what you would do with a sample on Earth. The samples we bring back from Antarctica, is there any life in this? Extract and sequence the DNA. But extracting and sequence the DNA works for life as we know it, and that's not what I'm looking for. So I say, if it's like us, then it's easy to search for life, but it's less interesting. In fact, I have zero interest in finding life that's just like us. And how could there be life just like us on Mars? I could have exchanged by meteorites. We realize that the solar system is not necessarily biologically isolated. So in fact, I would argue that the null hypothesis is that if there was life on Mars, it is related to life on Earth. And if it's alien, you've got to prove it, right? The assumption that just because you're from another planet, you're alien is no longer valid. But that's hard. How do we prove that something is alive if it's got a biochemistry that's very different from ours? And let me frame the question more directly. Here are a lot of examples of organic material. Oops. Here's the Tagus Lake meteorite, organic rich, lots of organics, including amino acids. Here's a laboratory experiment done in our lab, actually by a high school student, where we're trying to make organic material, the Miller-Urey experiment style. This was on Titan. And here is somebody's backyard, plants growing. Of these 3 pictures of organic material, only one of them is life. This is not life, this is not life, but the plant is life. And all the organic material in that soil derives from life, right? So how can we tell the difference? If we were to grab a sample of the meteorite and a sample of this garden, could we tell the difference between them? We think the answer is yes. And the poster child for this difference is in the amino acids. Both samples, all those samples that I just showed you, contain amino acids. Some made non-biologically and some made biologically. The difference is the biological amino acids are dominated by left-handed amino acids. Life uses only left-hand amino acids in its genetically coded proteins. It uses right-hand amino acids and other things too, but in proteins coded by DNA, only left-handed amino acids are used. So that means that in your body, the amino acids are vastly dominated by the left-handed version compared to the right-handed version. Unlike the meteorite, where they're 50-50, or a Miller-Urey experiment, where they're 50-50. This is a signature of the selectivity that life requires in its biochemistry. And you can make the argument from a evolutionary selection point of view, that life everywhere is going to have the same requirement for selectivity. Maybe not selecting the same. There's no reason why life couldn't have used D amino acids instead of left L amino acids. Just life, it's like driving on the road. You could drive on the left, you can drive on the right, but everybody's got to drive on the same side, at least within a community. Otherwise, it's chaos. Same thing applies. Life is more efficient and effective if it uses the same handedness in its biomolecules, right? So that's why life doesn't. And any deviation from that would be selected against by evolution. So this, in a sense, this molecular selectivity is a result of evolutionary selection. And let's just think of that in terms of analysis on another world. If we go to Mars and we find organic material and we find distributions of organics that are not just smooth and non-biological, they have unusual features in them. Say life on Earth has left-handed amino acids, not right, left-handed, not right-handed, left. That pattern is telling us that something is selecting those molecules. That something could be life. If it's alien life, it might have a different pattern, still have a pattern, but it's a different pattern. And that, I think, is the best way to search for life in the near term on other planets. So then the question is, what happens if we succeed? I gave this talk, or an early version of it, some years ago at Death Valley at the visitor center, and a little kid, I think she must have been like 4th grade or something, says, what are you going to do if you do find the second genesis of life on Mars? I said, well, that's a very good question. We spend a lot of time thinking about how to go to Mars, how to search for life, how to find a second genesis, but nobody's given any thought to what do we do if we find it? What does it mean? Does this change anything? We were like the dog chasing the car, unprepared for success. We had not thought through what would be the results of succeeding at what we're trying to do. So what would be the results? I think the first question would come up is, what is, at least to me, what is the moral status of alien microbes? Alien in the sense that they represent a second example of life. Now on Earth, microorganisms are treated with scorn. They score very low on any ranking of moral status in the animal kingdom that's based on pain, complex behavior, communication, etc. You kill them by the thousands every time you wash your hands or brush your teeth. And I encourage you to continue those heinous acts, right? But what if we find microbes on Mars and they are not just run-of-the-mill microbes? They represent the sole representatives of another completely different type of life, life 2.0. Their only representatives are microbial. I think that asks a new question that we've never encountered before. What is the moral status of not alien life? Normally you think when people ask that question, they're thinking about intelligent life, rational creatures. Kant would say they get all the rights that humans do. But what would Kant say about alien microbes? He'd say, kill them, who cares? Only rational beings get moral status. I don't like that answer. I think we should give them a much higher moral status. And in fact, the immediate recommendation that comes from that is that we should do our exploration on Mars in a way that's biologically reversible. And that is that if we find life on Mars, and it is a second genesis, we undo all the contamination that we have already done and resist from doing any further contamination, and instead enhance the life of that alien life. Okay, that's an interesting point of view, and it's consistent with the way we've done robotic exploration so far. We haven't done irreversible contamination of Mars. We've contaminated, but it's not irreversible. And we can also do that with human exploration. Human exploration could also be done in a way that's reversible. Now I want to move even further forward on Mars. We talked about the past, the present, the challenges of the present, searching for life, focusing on life. Those other challenges like the low gravity and so on, but the challenge from my point of view is a challenge of life, the future and life in the future. And this topic has a long history. More than almost 35 years ago, we wrote a big paper arguing that bringing Mars to life is an interesting topic and should be considered. And right now, there seems to be a resurgence of interest in this. In fact, there's just a paper out in Nature Astronomy led by Edwin Kite to make the case for more terraforming research. And as it happens, Astera an institute not far from here, not far from where I am, up in the Bay Area, whose goal is to support the creation of public goods to accelerate scientific and technical progress, announced its first residency selections, 6 residents. I don't know how many competed, but I bet it was a lot. It's a pretty choice residency. And 2 of the first 6 residents are related to Mars life. So that's, plus, of course, all the political mumbling about going to Mars and people saying we're going to go there to live. And Mars seems to be back on the agenda in a big way. And I think it's important that the wider community engage in this. In a sense, Mars is too important to be left just to scientists. It's got a lot of implications that are not just scientific, as I tried to just tease out here quickly with the question of ethics of a second genesis. So let's just look at terraforming from a simple point of view. It's just warming Mars up. You have Mars as it is today. You have Mars as you might like it to be. And you can just calculate the energy difference between the 2 states, the free energy difference. This is the first law of thermodynamics. There's no getting around it. If you want Mars to go from state A to state B, you've got to add this much energy. It's just fact of life, fact of the first law of thermodynamics, which is the most undisputable law in the universe. Coming close second would be the second law of thermodynamics, which we'll get to in a second. So what does the state need to be? You can go through what do plants need, what do humans need. Turns out that it's logical to divide Mars into 2 states, possible states, one suitable for plants, lots of CO2, not much oxygen, and one suitable for humans. Humans are much more picky about their atmosphere than plants, but we can list what's needed where you got to get there. So one big point in the terraforming discussion is there are 2 possible habitable states, one with and one without high oxygen, and address them both. High CO2, low oxygen, plants, insects, and worms. Some of my best friends are worms. I like plants, not so hot on insects, but it's okay. Low CO2, high oxygen, you've got people, which even better, but maybe not possible, as I'll argue. Okay, how do you do it? The only plausible energy source for terraforming Mars is sunlight, to dispel the myth that nuclear weapons can do it. And just a simple back of the envelope calculation shows that there's no nuclear summer for Mars. The total energy in all the weapons of the Earth, including Pakistan and India, adds up to something like 4 times 10 to the 19th joules. The sunlight hitting Mars every second is 10 to the 16 joules. So in about 1,000 seconds, just 1,000 seconds, Mars receives more energy from the sun than the combined nuclear arsenals of the entire Earth. We do not command energies on planetary scale. Dissuade yourself of that hubris. I agree, a bomb going off over your city would be a bad thing, but that's a tiny event compared to the size of the Earth or the size of Mars. And you can calculate how much energy is needed to evaporate CO2 on Mars. This notion you're going to bomb the poles and then create a thick atmosphere is just rubbish by about 5 orders of magnitude. And this is shown in the next slide. Here's Mars, in a sense, terraforming itself every summer. As the poles are heated up in the summer, that solar energy releases CO2 and it goes in the atmosphere and the atmosphere gets thicker. This is Viking data. The space between these 2 arrows is the amount of CO2 that would be released from the detonation with 100% efficiency. Forget the second law, 100% efficiency of all the nuclear weapons on Earth. This little space, it's in the noise of these curves that are occurring naturally. To terraform Mars, we need this curve to keep going up. Nuclear weapons are impotent in this regard. So it's got to be sunlight. So the logical calculation then is to say, we know how much energy it takes. We know how much sunlight is reaching Mars. What does that translate into years? If you could trap all the sunlight reaching Mars with 100% efficiency, thereby obeying only the first law of thermodynamics, how long would it take to terraform Mars? And the answer is surprisingly short. 7 years, 1 year, 17 years, 56 years for deep warming. These are timescales that are so short, I've easily passed them all in my own personal life, right? I'm roughly 100 years old if you round it off, right? I can easily understand projects that take this long. This assumes 100% efficiency. Every photon is trapped and used for warming. That satisfies the first law, but it doesn't satisfy the second law. The second law says you're never going to get to 100% efficiency. So what kind of efficiencies might there be? Warming, heating things up is thermodynamically easy. Efficiencies could be 10%. So the total time to warm, instead of being 10 years, might be 100 years. Again, that's how old I am. I get that sort of age. I can wait 100 years. I can start a project that will take 100 years to finish. But making oxygen, the efficiencies are more like 10 to the minus 4. So now it's 100,000 years. Now that's beyond the event horizon. That's beyond human event horizon. So I argue that just simple first and second law analysis argues that warming Mars, making it habitable for non-oxygen dependent organisms, is thermodynamically easy, and making it oxygen rich is hard. Now, this is a very simple argument, and it's subject to revision, of course, but it's going to be hard to change these numbers very much because you're working against process independent constraints of total energy and efficiency. Okay, how do we do it? The original suggestion is greenhouse gases. That's how we're warming up the Earth. And that would work on Mars. There are now suggestions for methods that could be more efficient that are being worked out, but the total amount of energy you need to do and the efficiency with which you need to do it will still constrain how long it takes to get there. For greenhouse gases, we've learned a lot about that. We have a lot of experience. One of the things I like about the greenhouse gas approach to warming Mars is we have a lot of experience doing it. We know what gases not to use. We know what gases are good. We want to avoid chlorine and bromine because they destroy ozone and so on. And there might, just might, be a way to couple the production of these gases to biological systems. There are organisms known that have enzymes that couple to these sort of exotic gases. So you might be able to get humans out of the loop by coupling in microorganisms to make the gases for you. Okay, we can calculate how much you'd have to add, and it's not ridiculous. The process could, again, take 100 years, and we have demonstrated it on Earth. The big issue with warming Mars, with terraforming Mars, though, is does it have the ingredients? Think of it as baking a cake. Thermodynamic argument shows that you have an oven, and the oven can bake the cake, and you don't have to wait too long for the cake to bake. But what we don't know is, do we have the ingredients to make the cake? What the thermodynamics just tells you is that if the ingredients are there, you can bake the cake. But we don't know if the ingredients are there. That is the big question. That is the big question. And some people have argued that makes terraforming impossible because the ingredients aren't there. But we don't really know that because these ingredients are in the soil below the surface. We know how much we need to make an atmosphere. We need CO2, water, and nitrogen. This detection of nitrates was done for the first time just with curiosity results. We are literally only scratching the surface of our understanding of these volatiles on Mars. And that's where research on near-term missions is going to inform the possibility of terraforming. Are the ingredients there? Again, we know that the thermodynamics works out. We know how to bake this cake. We just don't know if the ingredients to make the cake are present. Okay, so summary, is near-term terraforming of Mars feasible? CO2-rich Mars? Maybe. The physics works. The timescale is realistic. The big question is, are the ingredients there? The only way we find out is subsurface. Drill, baby, drill. I like that on Mars. Oxygen-rich? I argue no. The efficiency for oxygen reduction is too low by 3 orders of magnitude. Okay, how to think about it, how to make progress on this, I think is one metaphor I like, was suggested by Jim Graham. Think of warming Mars as going down a mountain. You start off where it's cold and frozen in the tundra, and you go down, and when you hit the tree line, that's when things really crank up biologically. So I'm doing a project with some colleagues in Poland, calculating when would the first tree be able to grow on Mars? How much warming, where, and where would the first tree grow? When we started, I suggested to them that it's probably going to be at the equator. That's where the first tree will grow. But it turns out that's wrong. The first tree grows in the southern hemisphere, in Hellas Basin, because the southern hemisphere, Mars has got an eccentric orbit. And so it has long, hot summers relative to the northern hemisphere. And so a long, hot summer can grow a tree. And so these are artist conceptions of what trees might be on Mars, what people might be like walking around on a Mars which has a thick atmosphere but no oxygen. The trees might get tall. Candace Elliott suggesting that the trees might go much taller in the lower gravity. Michael Carroll showing how humans would only need a mask to provide oxygen. They wouldn't need spacesuits anymore. Okay, I want to pull again the ethics in here. Mars exploration in general and terraforming in particular raised two completely new questions in environmental ethics. First one, terraforming, is life of more value than nature? That's a question that makes no sense on Earth because life and nature are the same thing. We use the words interchangeably. Respect for nature, respect for life. Say it either way, everybody understands what you mean. On Mars, nature appears to be lifeless. And certainly the rest of the solar system appears lifeless. Would it be of more value to have life there? I think the answer is yes. I am pro-life. I think we should have life on Mars. If I drive down the road and I see people with stickers that say pro-life, I go, yeah, pro-life. Let's terraform Mars. Life is the gift of Earth. And Mars, if Mars has the potential, we should share that gift. The second question is related to that. What is the ethical value of the second genesis of life? And as I mentioned before, this is again a question that we have no easy answer to from our experience on Earth, because we've never encountered a second genesis of life on Earth. We've never had to deal with an organism that is not related to us. So one of the arguments for preserving life and preserving nature is the codependency and the coevolutionary history of all life on Earth. That argument doesn't apply to a second genesis. The only argument that asserts an ethical value for a second genesis is a fundamental argument of richness and diversity as a value.
Hey, Chris.
Yeah.
We need a little time for questions.
Okay. All right. I'll just end with why do it all? Why do terraforming? Just because fundamental ethical principles related to diversity of life, utilitarian benefit, restoring life is a good thing. I'll end with a picture of Earth. And this is my intentionally blank slide at the end. Okay, great. I'm going to stop sharing, Kriyan, and we could go back to regular mode and have questions.
Okay, so I'm going to ask the first question or two, if you don't mind. First question is a fun one. Maybe if you might describe what is the Mars underground, and is there any relationship at all to Elon Musk's Mars fever?
The Mars Underground was a name given to a group of graduate students at the University of Colorado. The name was given by Leonard David, and there's a bunch of crazy grad students that were interested in Mars and the questions of terraforming, all the questions I raised here. And they all then went off to become professionals in space science, and most of them now work at NASA Ames. Myself and Carol Stoker and Penny Boston and Larry Lemke, folks like that. That's where the history is. Whether they had a direct influence on Elon is hard to say. I did meet him many years ago at a dinner party and gave this presentation about Mars, similar to the one I gave today. That was about 25 years ago.
Zubrin certainly influenced him. Was Zubrin part of the underground?
Yes, he was indoctrinated by the Mars Underground. He was in nearby Denver.
Well, he definitely influenced Elon. Okay, next question. What if we find life on Mars that could fit into the existing tree of life, but only if the root of that tree of life was like farther upstream than our current route on Earth life? So if it had RNA, but not DNA, or if it had a different but a related genetic code, as opposed to, say, unambiguous second genesis, which might have like right-handed amino acids or no RNA at all.
That's a very perceptive question. And Alfonso and I wrote a whole paper on that question. And that would be very interesting. And there's no reason to think that the last universal common ancestors I showed on the tree is the origin of life. There was certainly a long bit before that we don't see, and we can't see past. And if Mars was related to us and broke off before that, our at least universal common ancestor, it would show us a deeper common ancestor. It would be the outgroup still on the tree. Very interesting. Not a second genesis, of no interest to me personally. It doesn't take n to 2. N is still 1. What a drag. We need n to get to 2. My goal in life is to count to 2 and then retire.
Okay, let's see. Does anyone else have a question they'd like to ask? Because I have one or two more, but I don't want to dominate. Okay, Micah?
I'm curious, it sounds like you assume that all life will be organic. Is there a reason for that?
That's a good question too. The answer is we don't know. We don't know if life can have other forms besides based on carbon and living in water. That's how I would describe life on Earth. It's based on carbon and living in water. My approach is just practical. We know at least that life can be based on carbon and live in water. So let's go look at places where we see carbon and water. And that takes us to Mars, it takes us to Enceladus, and it takes us to Europa.
Let's take us to Titan.
Titan is next, because Titan is a lot of organics, but there's no water, but there's another liquid. So then you could say, well, can I take a tiny step? Could I expand my understanding one step and say life is carbon living in any liquid? Then we look on Titan, and we're going to do that. To my amazement, the Dragonfly mission includes as one of its level 1 science objectives the search for life that would be carbon-based and living in liquid methane. And if we find it there, that is obviously a second Genesis. That would be a wonderful example of N equals 2.
Okay, so that's a great lead into my next question, which is in terms of upcoming missions, which are, say, on the books, rather than just simply being about, what are the most exciting astrobiology-related missions, search for life-related missions in your mind?
2 upcoming. One is Europa Clipper. On its way now, we'll orbit Europa, and we'll have the capability to detect organics on the surface. And even if we're lucky, if there's biological organics, it might be able to see them, because it collects fragments sputtered up into space. It collects it with an instrument that destroys the molecules and looks at the fragments. And we just might luck out and find evidence of life there on Europa's surface. It's a long shot. You'd really like to get into the ocean, and we don't know how to do that. The second, and that mission will reach Europa in 2034, roughly. The next mission of interest is Dragonfly, which will go to Titan and search for organics and characterize the organics that are there, and maybe find evidence for organic life in liquid methane. I will do it 2 ways. One is organics, and the other by looking for hydrogen variation. Again, it will arrive in 2034.
Okay. What about missions to Mars? Are we sending, or is anyone sending anything that has chips on board to look for varieties of molecules or all this kind of new technology that wasn't around for Viking and Phoenix?
We'd like to send another mission to Mars, but right now there are no approved missions. You said approved. There are no approved missions to Mars being planned. The great hope was the sample return would bring a sample back, and we'd use all this wonderful new technology on that sample. That is in danger right now. I think it's on the chopping block in the budget process. So right now, Mars, the answer to your question really is just Europa and Titan, and wait till 2034. So all of you need to be around in 2034. We'll have another seminar. We'll talk about the results from Clipper and Dragonfly. So stop smoking, go running every day. You've got at least 10 more years to stick around, and I'll meet you then. We'll make a calendar invite.
Cool. We're on. Another question. This is my paraphrase of a question in the chat. Where do you come down on UAPs and alien visitors to Earth? I know that's an out of left field question, but people are interested.
Yeah, no, I get the question a lot, and it's not a ridiculous question. It's just not a question that interests me. However, if we do contact intelligent aliens, either through SETI or on the White House lawn, my favorite movie is still The Day the Earth Stood Still. The first question I'd ask them is, are you DNA based? Do you map on our tree of life? If no, then I'm interested in you. If yes, get out of here. If not, then I'm not interested. I'm still looking for 2.
That's cool. That's cool. All right. Does anyone else have a last question? Otherwise, we might wrap this up 2 or 3 minutes early so I can catch my ferry.
Oh, yeah.
I think Luke had a question.
It's worth the time. It was just that earlier in the talk, you had said that you don't want to use chlorine or bromine as a greenhouse gas heating accelerant on Mars because of the threat to the ozone layer. But my understanding is that Mars has a really scant little bit of ozone in the atmosphere in the first place. Like
Right.
what are you trying?
Yeah, but you'd like it to be less scant. And so if you introduce a thick atmosphere with higher CO2 or with oxygen, no, the ozone should go up. So you don't want to do anything to destroy it, because the ozone does have the beneficial effect of blocking out ultraviolet light, particularly UVC, which right now is not blocked on Mars. And it's the main reason Mars has not been irreversibly contaminated, because that UVC is sterilizing, putting the entire surface in a sterilizing bath, which is fine now. It means that any contamination released from our spacecraft is quickly killed. But once we're there and we want to live there, then we want to get rid of that. So having an ozone layer is a good thing. And so you would want to use versions of the greenhouse gases or a warming method in general that doesn't this negatively affect the production of ozone, which is photochemically produced.
Hey, Micah, you got one more?
Yeah, I was wondering if you could expand a little bit on what the constraints that causes O2 production to be so inefficient. Is it a physical constraint, or if we don't know how to make it efficiently?
We don't know how to make it efficiently. All efficiency constraints are due to lack of knowledge of a process that could be higher efficient, because the only real limitation is the first law. But right now, the only way that we know how to make oxygen on the scale of a planet is with biology, and that's plants. And so if you look I got my number 10 to the minus 4 by looking at the global efficiency of Earth's biosphere. What is the efficiency with which Earth's biosphere turns sunlight into oxygen? And the answer is about 10 to the minus 4. Now that, I think, is an optimistic assessment of the efficiency for a biosphere on Mars, because Earth's biosphere is rich and thick. And the Martian biosphere is going to be pretty thin and cold, more like a cold desert, even after it's warmed a lot. So it's hard to imagine that the efficiency of a biosphere on Mars is going to be better than the average efficiency of the biosphere on Earth. So it's a hard problem, and it has to be tackled in the efficiency area. Yeah. So that's why I tend to be pessimistic about making an oxygen-rich terraformed Mars soon.
Okay, Chris, this has been wonderful and all we'd hoped for and more. And I want to thank you for taking the time. And I want to tell everybody that if you have friends who missed this, the video should be up. I don't know, Lydia, how long does it take these videos to go up on YouTube?
Maybe a week.
Okay. So yeah, and we got more great speakers to come. Lydia and I are gathering up the next batch. And if you all know of someone who you think should address this group in broad areas of aerospace, planetary science, spacecraft engineering, exploration, the future. Yeah, let's hear from you. And I want to thank everybody for coming, and I'll see you next time.